Okay, so our next speaker is Saskia Vries from Allen Institute for Brain Science. Her talk will be about a large-scale standardized physiological pipeline reveals functional organization of the mouse visual cortex.
Great. So thank you so much for the invitation to speak and for everybody who's done a lot of work to still make this this meeting happen, given our current circumstances. I appreciate kind of everybody pulling everything together at the last minute to be able to do this virtually. So yeah, so I'm going to tell you about another project at the Allen Institute that was happening in parallel with what Nathan was just telling you about, to look at the physiological activity of these neurons in the mouse cortex. And let's see, there we go. And in Nathan's talk, he had motivated some of why we're interested in the cell types by in terms of where we want to understand about how the architectures of the circuits are formed and how these circuits are, how they might support different types of computation in the cortex. And this is really some of the questions that motivated our efforts to conduct a survey of functional responses for different cell types in the visual cortex. We wanted to be able to ask questions about how visual sensory information and visual information is being transformed by the circuits, how visual information is being represented, and whether there's differences between different cell types in different layers or in different parts of the cortex in terms of their visual responses. And so to address this, we created the Allen Brain Observatory, which is essentially a survey of physiological activities that we measured using 2-photon calcium imaging. And so this is an example of one of our imaging sessions. We're using the same transgenic tools, these different Cree lines, to drive the expression of a calcium indicator. In this case, it's GCAMP6, that we're driving in a particular subpopulation of neurons. And then we can image the activity of these neurons that you can see here while the mouse is watching an array of different, a battery of different visual stimuli. So you can see here we've got a few hundred cells that are labeled in a given field of view. And we can see that different cells light up at different times. And we can start to ask questions in terms of what's the stimulus pattern that's present when any given cell or any group of cells are active. You can see in this video, what I'm showing here in this middle panel, the second panel is the calcium activity. On the far right, you can see the stimulus that we're showing to the animal. And in this session, I'm gonna just play this again. We've got some noise stimuli as well as some natural movies. And at the same time, we are capturing the running activity of the mouse. So the mouse is awake and it's on a running disc, and there's times when the mouse will run and other times where it'll stand still. There's no task involved in this component. In this experiment, it's completely just passive viewing. And so different mice will run at different times, but there's nothing really driving that running activity. We also can track the eye position of the mouse. And so you can see this red dot over here corresponds to the position of the mouse's pupil during the experiment. And during a given session for these mice, when they're head fixed, they tend not to move their eyes very much. If you were paying attention, you'll see that there are some subtle movements back and forth, but they're fairly minimal and fairly small during these experiments. So we use this type of calcium imaging to capture the activity of populations of neurons. And we've done this using a high throughput pipeline where we have dedicated teams that kind of carry out each step of this experimental procedure. So we have our transgenic mice that are being generated at the Institute. The mice undergo surgery in which we create a window in the skull, an optical window that gives us access to the cortex, as well as we attach a head post so we can head fix the mice. We then hand it off to a team that does intrinsic signal imaging that can create a map of where the different visual areas are located on the surface of the cortex, which allows us then to target our imaging in the later stages of the experiment. Mice are habituated for a couple of weeks so that they're comfortable being head fixed and being on this running disc. And then we collect the calcium imaging data, the in vivo 2-photon imaging. For each group of cells, for each field of view, we image the same field of view 3 different times, which allows us to use a larger set of stimuli. So we come back on multiple days to image those same cells, and we'll collect data from 2 or 3 different fields of view from an individual mouse. Once we've finished collecting the calcium imaging data, the mice are euthanized and then go through a histology step of serial tomography imaging that gives us allows us to confirm that the expression pattern of that particular transgenic line is what we expect it to be, that the right cells are being labeled, as well as to make sure that there aren't any kind of brain abnormalities or deformations that sometimes occur. This type of pipeline, because we have separate teams at each stage of the pipeline, we have to use a lot of engineering. And so this is kind of where the Institute with our large teams kind of can sometimes, really shine, is that we've developed a lot of hardware that allows us to keep all of the instrumentation across each stage of this experiment aligned to a common coordinate system, so that when the surgery team or the intrinsic imaging team can define coordinates that we want to use to target for the calcium imaging at later stages, they simply have to hand off a set of numbers to the other team that they can use to guide the instrument to the right location. The pipeline, this type of standardization also enables us, it allows us to run a lot of mice through this experimental pipeline, but it also allows us to define strict quality control criteria for each stage of the pipeline, that we can make sure that the animals and that the data that we generate at the end of this all meet the same standard for quality, for health, for the animals, and for the quality of the imaging data. So over about a year and a half, we collected about 1,400 hours of imaging from over 240, 250 different mice. And so we have a lot of movies of calcium activity, a lot of movies of eye movements. And so we, in addition to a data collection pipeline, we developed a data processing pipeline that carries out in an automated way the image registration, the motion correction, the segmentation of the different ROIs, the different cells in each image, to do the trace extraction, demixing, neuropil subtraction, that we then, in order to be able to then extract the activity of all of these individual neurons, that we can use in our analysis to relate this activity to the visual stimuli and the behavior of the animal. So in this time, we collected data from over 63,000 neurons. So this table just gives you a summary of how many neurons and then how many experiments were collected from each of the visual areas, and then each of our different transgenic lines. So we collected data from 6 different visual areas, the primary visual cortex, and then 5 higher visual areas, and then from a battery of different Cree lines, many of which they differ in terms of whether they're excitatory or inhibitory. So we've got our 3 major classes of inhibitory neurons here. And then we also see difference, they also differ in terms of which layers we find some of these excitatory neurons in, some of them labeling specific subpopulations within a layer. To unpack this just in a slightly different way, here I'm showing you the surface of the mouse, the dorsal surface of the cortex. Here we've got the primary visual cortex. The barrel cortex is up here in the top corner, auditory cortex over on the left. And so highlighted in green are the 6 different visual areas that we've collected data from, primary visual cortex, and then 5 of these higher visual areas that surround it. These are the Cree lines that we've used, the 14 different Cree lines. We've got these sparse inhibitory ones, VIP, somatostatin. We did collect data from parvalbumin, although we haven't included this in our analysis for a variety of reasons. So I'm not going to tell you about parvalbumin, but we do have data collected from that. And then all of these excitatory lines, some of the lines are pan-excitatory, so they're expressed across all of the layers, whereas others are restricted, as I mentioned before, to particular sublayers or to particular layers, and then sometimes within that to subpopulations within those layers. And then we used a battery of different stimuli. These include some kind of the traditional stimuli, such as these sinusoidal gratings that are either drifting in different directions, or flash static gratings that have different orientations or spatial frequencies. We have a locally sparse noise stimulus, which has white and black spots that are flashed in different locations across the monitor, as well as a set of natural images and natural movies that we use. For the natural movies, we've taken a couple of clips from the opening scene of Touch of Evil. And we have one clip is a 30-second clip that we repeat in every session. So I mentioned before that we image the same group of cells across 3 different sessions. And that's to allow us to show a lot of these different types of stimuli as much as possible. And so the only stimulus that gets repeated across all 3 sessions is this 30-second natural movie clip. We have 2 other movie clips, another one that's 30 seconds and another one that's 2 minutes long. So it has a longer duration, more features involved that are only shown in one session each. And so this allows us to really then be able to create this survey. Let's get rid of that. No, don't do that. This survey of visual activity, of physiological responses across these areas, across all of the different layers of cortex and within different subpopulations of neurons, using a range of different stimuli to really allow us to dig into these questions of how this information is being represented, how the visual information is being transformed, and what the type of computations are being carried out by this cortical circuit. So I can show you an example of one cell. As Nathan was mentioning with his data set, all of our data is available for anybody to use. And so you can go to our website and see visualizations of the data. I provided the link here, but I'm just going to show it to you in the slides here. But we also have a software kit where you can download the data and analyze it that gives you access to all of these data that we are describing. So this is one example cell. It's an excitatory cell in layer 5 of V1. And I'm going to show just quickly the responses of this cell to our different set of stimuli. We use some visualizations that on the website are fun, dynamic visualizations. They take a little getting used to, and especially when they're not dynamic, but this is a representation of the response to our drifting grading stimulus. So this is a sinusoidal grading that moves in 8 different directions. And these directions are represented by the different arms of this plot here that we call a star plot. And it also, the gradings move at 5 different temporal frequencies or speeds at which the gradings are moving. And these are represented by the rings of this star plot with the slowest temporal frequency in the center and the fastest temporal frequency on the outside. At the intersection of the arms and the rings, you'll see a collection of dots. Each dot represents a single trial of that condition. So for a particular direction at a particular speed, we've got 15 different trials. And each dot then is colored by the strength of the cell's response for that particular trial. So for this particular cell, we see that there's a lot of strong responses when we've got horizontal gradings that are moving up at about 4 hertz. There's a few responses when it's moving down, but they're much weaker. And we don't see any responses when it's moving, say, to the left or to the right. There's data there, but because the responses are so weak, those dots show up as white, and so we don't actually see them. And so you can kind of quickly get a representation of the orientation and direction selectivity of the cell, the temporal frequency, as well as some indication of the reliability of the cell's response trial to trial. A similar visualization summarizes the response to our flash static gradings. Again, the gradings are flashed at different orientations represented by these arms, and now at different spatial frequencies or periods of the grading, where the lower spatial frequencies, the thicker gradings are in the center, and the higher spatial frequencies or the finer gradings are at the outside. And so again, for this cell, we see that its strongest responses are when this grading is essentially horizontal, maybe a slightly angled off horizontal at some intermediate spatial frequency of about 0.08 cycles per degree. We can look at the response to our locally sparse noise and find the positions on the monitor where we see a large evoked response when we either have a white spot up here in this top plot or a dark spot. And so for this cell, we see that there's this region on the left side of the monitor where the cell responds when we have white spots. There's no subfield where we see a response to the dark spots for this particular cell. So we have this on receptive field for this cell. This plot here represents the response to our natural images. We have a set of 118 different natural scenes that we show to the mouse. And the responses for each scene are represented by one ray of this plot. We call this actually the corona plot. It has nothing to do with our current virus that's going around, but because it looks a little bit like a crown. Again, there's 50 trials for each image that gets presented, so each dot is a single trial. And so you can see for this particular cell, there are 4 images that drive fairly reliable and fairly strong responses. I've shown these images up above, so you can start to think about what features these different images might have in common. And finally, I'm showing a response for one of the natural movies that this cell responds to. This is essentially a raster plot that we've we've wrapped around in a circle where time goes around it like a clock, starting at the top. There are 10 trials, 10 repeats of the movie. And so these are the 10 rows here that are in red. The outer ring in blue is the mean across those 10 trials. And you see, again, there's these kind of a couple different time points during the movie where we see a reliable response across these different 10 trials. And we can look just here. You can see one of those moments in the movie where this street organ, this hand or whatever it's called, gets pushed into the image that we see. And so we can start to think about what these different images have in common. And I get the sense, especially from this movie, if you take this receptive field here, this spatial receptive field, that it likes light things over here on the left side of the monitor, and here you've got this bright street organ getting pushed into the scene. That might be the thing that's driving it. Maybe that's also the back of this tiger, or the contrast of below this wing that's eliciting the responses of these neurons. So that's one example cell. We've got 60,000 of them. And so you can imagine that we see quite a bit of variety in the responses that we see. This is a few more examples. This top row is the cell that I just showed you. On the left, I'm showing both the fluorescence trace, but also the extracted events. So one thing I should just mention is that all of the analysis that we've done, that I'm telling you about today, has been using the extracted events rather than the fluorescence trace.
Oh, put a jacket on.
You can see the relationship between those two over here. So here are a few more examples where we see these types of diverse responses with direction selective or less. This is a somatostatin cell where we see that it responds pretty equally in different directions, but it does show nice, robust, reliable responses to different gradings or different images. In the movies, you can see these really nice, robust responses here. And then our VIP neurons, these are the other inhibitory population we've been looking at. And these show some really interesting different types of responses. One of the things that's interesting with VIP cells is that they don't tend to respond to our full field high contrast drifting grading stimulus. And you can kind of tell that in this plot where we don't really see any cluster of dots the way we do for these other neurons. In fact, many of our VIP cells are actually suppressed by this grading stimulus. But we do see that the VIP cells show responses to a lot of different images, whereas most excitatory cells and this somatostatin cell respond to just a few images. The VIP cells respond more broadly across our set of images. We also see consistent responses to our natural movies that we show, as well as really nice and actually surprisingly large receptive fields that we can map with this sparse noise stimulus. So we can start to ask some questions about these responses, and one approach that we can take is to derive classic metrics about visual physiology. So we can quantify the strength of the direction selectivity of the cell. That's how much it prefers when the grading moves in one direction, its preferred direction compared to its null direction. This particular cell has a direction selectivity of 0.7. A selectivity of 1 means that it only responds to the preferred direction. A selectivity of 0 means that it responds equally to the preferred and the null directions. And so this cell is actually fairly strongly direction selective. Similar thing for orientation selectivity. We can derive metrics in terms of the preferred tuning properties for temporal frequency or spatial frequency or orientation, the area of the receptive field. We can measure the sparseness of the response to the images. This is essentially a metric that quantifies how many of these images does the cell respond to. So this is a fairly sparse response that it responds to about 4 images here. So this corresponds to a fairly high lifetime sparseness, or the reliability of the responses across these different dimensions. And so we can derive these metrics for all of our neurons and start to look for patterns or look for differences in these types of response properties. And so in order to do this, when we've got this type of large data set, we need to find ways to visualize this that allow us to make some comparisons quickly. And so we developed a visualization that allows us to compare the responses across these different areas, and then again across layers. And we represent each area here with a circle, and we've arranged them kind of roughly how the areas are represented on the surface of the cortex. So we've got V1 in the center and then the 5 higher visual areas around it. We call this configuration a paw, like a paw print of a bear or something. And then we can repeat this paw for each layer that we've collected data from, so layers 2, 3, 4, 5, and 6. And then we can color each of these circles using a heat map that can summarize the average value of whichever metric we're thinking about. So for example, we can look at the direction selectivity of these neurons and compare how the median value of the direction selectivity index for in each of these areas and each of these layers. So here's this example neuron that we've been looking at just to remind you what this metric is. And what we see when we start to compare the direction selectivity is that we see very high direction selectivity in layer 4. Actually, across all of the areas, we get the highest direction selectivity in layer 4. And this is particularly true in V1. And that's a little bit surprising to us. I think we expected, based off of decades of research in other mammals and cats and primates, that layer 4 of V1, where the thalamic inputs are coming in, usually has the least direction selectivity information, and that direction selectivity is computed kind of in subsequent stages of the cortical circuit. But in the mouse, we actually see something different, where we have very high direction selective information right away in layer 4 of V1, and that this actually gets weaker as we move away from this. We can dig a little deeper. So one thing I should note in these paw plots is that we only use a single Cree line for each layer. If you remember the table that I showed you several slides ago, not every Cree line was sampled across each area, across all of the areas equally. And so to do a fair comparison across the areas, we limit for each layer to a single Cree line that was sampled uniformly across the areas. But within a particular area, so in this case within primary visual cortex, we can look more deeply at the distributions for each Cree line. And so, what we can see here now is these different types of Cree lines that are found in the different layers. And so, for instance, this elevated direction selectivity in layer 4, we see is not unique to this particular Cree line, the Bohr B cells, but is actually consistent for all of the excitatory cells in layer 4. Even these panexcitatory cells that we've imaged across multiple layers, we see that in layer 4, they have the highest direction selectivity. Then we've got our inhibitory cells that have very low direction selectivity. I pointed this out for the somatostatin cells earlier, that we see they don't tend to have direction selective responses. So we can take this approach and compare a lot of different metrics. And here I'm just foregoing the paw plot, but just looking within V1, we can ask questions of, do we see large functional differences between our different Cree lines? And so I'm showing a number of different metrics here, the direction selectivity that we just looked at, preferred temporal frequency, preferred spatial frequency, the receptive field area, and the lifetime sparseness, which is how selective the cell is for how many images it responds to from our natural image set. And it's a little bit of chaos because we have so many Cree lines. But if we start to kind of focus in, so if we look just at the excitatory one, so I've grayed out our inhibitory populations, there are some differences, but by and large, we see that across our excitatory neurons, the differences in their response metrics are fairly small. I can point out some differences here where the receptive field area of these different subpopulations within layer 4, the receptive field sizes are different, and this corresponds to changes in the spatial frequency tuning of these neurons. And this makes sense. Those two kind of go hand in hand. But other than that, the differences tend to be fairly nuanced and subtle. If we ignore the excitatory populations, though, and focus in on the inhibitory neurons, there we start to see much bigger differences. And I've already mentioned earlier when I showed you a few example cells that we see big differences in terms of the VIP cells that don't really respond to our large full field gratings. Here we're just showing for the few that do respond, they show very different tuning properties than we see for the somatostatin cells in terms of they tend to be more direction selective than those somatostatin cells. They tend to have slightly different tuning properties. But as we move into the receptive field areas, again, the VIP cells have actually remarkably large receptive fields when mapped with this sparse noise stimulus. And really, and I showed this in the example, they have very low sparseness for the natural images. So when we show a lot of these different images, they respond quite broadly to a number of the images as opposed to the somatostatin cells and the excitatory populations that have very sparse responses. So we actually see really big differences in these inhibitory neurons that are suggestive of very different roles in the cortical circuit for these two populations. All right, so one of the other questions that we really wanted to be able to get into with this data set was, can we develop models that will predict the visual responses? How well can we predict these visual responses? And in particular, we wanted to test what we can refer to as the standard model for the visual system. And so, the visual system has been studied for many decades, going back to Hubel and Wiesel, where people began measuring what they call receptive fields and defining stimulus features that drive the responses of different neurons. And over this time, we've developed kind of this standard view of how, particularly the early visual system, what these neurons respond to, where there's some sort of spatial and temporal feature that drives the response of a cell. And when that feature is present in an image, it's going to evoke a response. So this is an example for a retinal ganglion cell or for an LGN cell, where you've got a concentric on surrounded by an off. So it's on center, off surround. And when you've got the spot of light like this, you're going to evoke a large response. But if you have the opposite stimulus, say a spot of dark on a light background, you don't see a response. And so you've got this type of rectified output based off of this spatial temporal feature. And then as you move into the cortex, these become, rather than concentric, you've got these elongated areas of flanking on and off regions. And again, if you've got an oriented bar that aligns with this, you'll see a strong response, but anything that is kind of out of phase will not evoke a response. And so just to reiterate this, if our expectation for this type of simple cell, if we show a grading that aligns with the same orientation, we've got the dark flanked by the light, we're going to evoke a strong response from this cell. But if we were to show it some other stimulus, say this natural image of this flower, where the stem falls in the same location as that oriented receptive field, we also expect to see a response to this type of image. Then there's also, in addition to these simple cells, we see in primary visual cortex these complex cells that show a phase invariance. And so now we move from having this type of rectified output to having a quadratic rectification, so that regardless of the phase of the grading or other features, you'll still see a response if it's light with dark or dark with light, but as long as it's got that orientation and spatial frequency tuning and all of those things. And so this is the model that's emerged in the field for a while. But in the last little over a decade, people have kind of pushed against this model a little bit for a variety of reasons. And one being that when people do try and test this with more naturalistic stimuli, we see kind of greater differences in the responses that are evoked. But there's also a sense that we might be biasing our impression of the visual cortex based off of our experimental methodologies. And so oftentimes when you go in with a single electrode and you're recording from a single cell, I mean, first of all, you're not going to record from a cell that's not responding really robustly to the stimulus that you're showing, right? You're going to keep hunting for a more exciting cell to record from. But then you're also going to optimize the stimulus for your cell. You're going to change the orientation of your grading, or you're going to select different features to show to that cell to really get the best response that you can. And so one of the problems that emerges is that for every cell that gets recorded from every different group, the responses are always to a different set of stimuli that have been optimized in different ways. So one of the things that people have been asking for a while is to create some type of survey, a database that allows us to more equitably compare the responses of different neurons and different types of neurons within the visual cortex. And so in many ways, this kind of summarizes the motivation for why we wanted to create the Allen Brain Observatory, was to really be able to reduce some of our experimental biases in recording quite broadly from lots of cells simultaneously. None of the stimuli get optimized for individual cells because we've got a few hundred cells at a time that we're imaging. So we can use our data now that we've collected it to test this standard model. How well does this type of model with a spatial temporal filter followed by either a linear or quadratic nonlinearity, how well can that predict the activity of the neurons? So we developed, so we use this model. We have a 3D wavelet projection where we've got spatial temporal wavelets that at different locations and sizes and spatial and temporal frequencies that get followed both by linear or quadratic nonlinearities. And these all get added together, and then we can train the data to train weights for the different wavelets so that we can find the wavelets that are most predictive for individual cells. We either train this data and test it either using the natural stimuli, these are our images and our movies, or using the artificial stimuli, the noise and the gratings. And so we can both train it and test it on these different stimulus types. Also, I should note we include the mouse's running speed in this model. As we've known for the last decade, the mouse, the running activity of the mouse can modulate the visual responses of the neurons in the cortex. And so we want to be able to include this effect in our predictions. Then we can start to compare the predicted output with the measured response. And so this is, I'm summarizing here again with this paw plot and then looking within V1 across the Cree lines, where the heat map here is showing is the median model performance. So this is just the correlation of the predicted response to the measured response. And this is just for the natural stimulus. So we both train and test with the natural stimulus where we've done a 5-fold cross-validation of that performance. And honestly, the performance isn't that great, right? Our median value is about 0.25 in layer 2-3 of V1, which is probably the best performance that we see. We see lower performance outside of V1 and in these other layers. Layer 5 looks pretty good as well, but that's not really saying much when that median value is pretty low. And again, looking across the Cree lines, we still see fairly low performance with most of our populations kind of centered around 0.2, a little bit better in layer 5, and actually slightly better performance for our inhibitory neurons. Now, both VIP and somatostatin cells, they show really robust visual responses, but they also are strongly modulated by running, more so than the excitatory populations, and so that could be a key component to their model performance here. But if we just look at all of the cells now, and here what I'm comparing is how well we can predict the response to the natural stimulus, and then how well we are able to predict the response to the artificial stimulus. And this is a density plot, so the color here corresponds to where the bulk of the neurons are. And as we saw already, that the median value for the natural stimulus was even in the best kind of layer area was only about 0.25. If we're looking across all areas and layers here, we usually see that the bulk of the data here is actually quite close to the origin, right? Very close to 0, 0. Cells tend to be better predicted in response to the natural stimuli than the artificial stimuli, but by and large, we don't see great model performance. Now, that isn't to say we don't see any good models. So this is an example of a neuron that's really well predicted. We have R value of almost 0.8, both for the natural and for the artificial stimuli. So this would be a cell that's somewhere out here. But a lot of the neurons we see are either well predicted for the natural or well predicted for the artificial, but not seeing good kind of performance kind of for the other stimuli. And so this is a little, it was a little disheartening when we first were seeing this, because we were hoping to be able to do a little bit better than that. But one of the things that you know, in order to be able to predict a response, you have to have a reliable response. You really have to have good evoked activity. And one of the things that we notice in the data, and this is something we actually saw very early on, but really didn't kind of put it together until a little bit later, is that neurons tend to be somewhat selective in terms of which stimuli they respond to. Not all neurons respond to all the visual stimuli. The examples that I showed you earlier in my talk are neurons that I'd selected intentionally because they do show nice responses across all of the different stimuli that we show, the gratings, the movies, the sparse noise, all of these different types of stimuli. But most of the neurons we find are really robustly responsive to one stimulus and don't respond that well to other stimuli. This is an example of one of our imaging sessions. I don't actually remember where it's from, but I'm just showing the activity of 50 neurons from one imaging session. This is the entire hour-long imaging session, and I've colored in the different epochs of the stimulus presentation. And so one of the things that we've done is we show different stimuli in different sessions, but then within a session, we interleave the different stimulus types among each other so that we've got 10 minutes of static gratings, then followed by 10 minutes of natural scenes. And then we come back and show more natural scenes and more static gratings. And we've got some spontaneous activity and some natural movies kind of interleaved as well. And what you can see just looking at this raster plot of activity, you can see these neurons like this example here, just below the number 40, that's really active, really has a lot of activity while we show the static gratings. But then when we switch to the natural scenes, we see that the cell really doesn't respond very much at all. But when we come back, 20 minutes later and show more static gratings, the cell again responds quite robustly. So it's not like we lost the cell or that it died halfway during the imaging session, but it's really selective for this particular stimulus type. We see other examples here of cells that respond really nicely to the natural movies. Again, the movie gets repeated 10 times, and so we see this periodic response where the cell responds to some feature of the movie every time that it comes around. But then when it switches to the natural scenes, which have the same spatial statistics as the natural movies, but are different features and don't have any temporal correlations, but now the cell doesn't, is pretty inactive. You can see that here as well. So we find that cells are active for different stimuli, that they respond a lot to different stimuli. And so we wanted to look at this more quantitatively and ask how similar, like if a cell responds to one stimulus, is it going to respond to another stimulus? How much can we learn about its response to another stimulus from one? And so what we looked at was the correlation of the reliability of the responses, which we compute based off of the percent of responsive trials. So for instance, with the natural scenes, there's going to be one image that will drive the largest response for a given cell. And we can ask, of the 50 times that we showed that one image, how many of those trials did the cell have a significant response? And we determine the significance comparing each trial to a distribution of activities pulled from the epoch of spontaneous activity over here. And so we can determine what percent of trials of its preferred condition of the natural scenes do we see a response. And we can ask the same thing for the drifting gratings or the static gratings. And so what I'm showing here is a heat map of the correlation of the percent of responsive trials for its preferred condition for the natural scenes and the percent of responsive trials for its preferred condition for the drifting gratings. That would be this location here. And I'm doing this across all these different stimuli. We've got the static gratings, drifting gratings, natural scenes, and then all of these are different natural movie clips. And remember, I told you earlier that there's one clip that gets repeated across all 3 sessions. So natural movie 1 in session A, B, and C. And so the correlation across these different presentations of natural movie 1, these give us a kind of an estimate of the variability that we expect from imaging the same cells on different days, right? So it's the same cells responding to the same stimulus, but at different times. And here we get a correlation of a little above 0.5. But everywhere else, we have actually very low correlation, with one exception being between the static gratings and the natural scenes. These are the 2 flash stimuli that we show. Knowing that a cell responds really robustly, really reliably to, say, the drifting grating stimulus, what that means is that it tells us very little information about whether that cells will respond reliably to, say, the natural scenes or the natural movies or the static gratings. It doesn't mean that the cell won't respond to those stimuli. It tells us that we can't make a guess as to whether the cell is going to respond to those different stimuli. And so we wanted to dig into this again a little bit more, and so we did a clustering on this this joint reliability. So now we can take the percent of responsive trials for each of these stimuli: drifting gratings, static gratings, natural scenes, and natural movies. Because we have these multiple different natural movie clips, we basically take whichever clip evoked the most reliable response. That's the value that we use. And we used a Gaussian mixture model to do a clustering analysis. And we find these 30 different clusters that what we're representing here is the mean percent responsive trials for each of the 4 stimuli. So if we zoom in on one of these clusters, you can see that it has high reliability for the drifting gratings and the natural movies, and lower for the static gratings and the natural scenes. And the way that I set the color bar on this plot is that we took the cluster that had the lowest mean percent of responsive trials across all of the stimuli, and we used that to set a threshold. It's 1 or 2 standard deviations above that mean, and that's our threshold then for saying if there's a greater percent of responsive trials, then we'll color that red. We consider that to be responsive, and if it's below that, it's blue, and we consider it to not be responsive. And so then what we can do is group these clusters into classes based off of which stimuli are they responsive to, right? So here we've got a class of these 2 clusters where we have reliable responses to the natural movies, but to none of the other stimuli. And here the same for both drifting gratings and natural movies, but not the static gratings and natural scenes. So we get a number of clusters that fall into this class. Now, one thing to note is that, as we saw in the slide before, there's still a low correlation across these stimuli. So some of the cells, some of the clusters in this class, you've got equal reliability to the 2 stimuli, whereas others you see it's very reliable to the drifting grating, but not super reliable to the natural movies. So there's still a lot of variability within these classes, but we're able to create these larger classes. We repeated this process 100 different times with different starting conditions, and could then look at how many neurons fall into each of the classes that we find. And so there's a couple of things that really pop out. So our largest class, by and large, are these cells that don't respond to any of our stimuli, don't respond reliably to any of our stimuli. 35% of neurons fall into that class. Our next biggest one is this blue bar, which are the drifting gratings and natural movies, the cells that respond to both of those stimuli with some reliability. And we think that these cells are kind of motion sensitive cells, because both of these stimuli have temporal correlations, they have motion features. The natural scenes and natural movies is the next biggest group, these clusters here. And these cells, what we think is probably the key feature is the spatial statistics of these stimuli, that having kind of these naturalistic 1/f spatial statistics are really key to driving these neurons. And then our group over here in green, these are the neurons that respond to all 4 of our different stimulus types reliably. With some reliability, and that only makes up 10% of the neurons in our data set, which it seems was lower than I expected it to be. I think it's really important to note that there's a number of possible combinations that we just don't see. For instance, we don't have any neurons that respond to, say, the drifting gratings and the static gratings, but none of the natural stimuli. Or we don't have any neurons that respond to drifting gratings and natural scenes, but not the natural movies or static gratings or things like that. So there are possible combinations that just don't exist, but that most of the neurons fall into some of these larger classes.
5 minutes.
Thanks. We can now compare how these classes relate to the model performance that we were looking at before. And it makes a lot of sense what we find, right? So if we look at the neurons that fall into this large group of none cells that don't respond reliably to any of our stimuli, these are the neurons that really create this bulk of the data down here right at 0, 0. And this makes sense. If a cell's not responding reliably, we're not going to be able to predict that unreliable response, right? And so the bulk of our data that falls down here in this corner is really because these cells just aren't being driven by the set of stimuli that we're using in these experiments. the cells in our natural class, the natural scenes and natural movies, these are really well predicted for natural stimuli, but then aren't well predicted for the artificial stimuli. Again, if it's not responding reliably to those stimulus classes, then we're not going to be able to predict it. So that also makes a lot of sense. And then the cells that fall into our all category, that they respond reliably to all of our stimuli, these are the cells that are well predicted both for natural and artificial stimuli. Our median value here is about 0.4 for both, right on unity. So for both the natural and the artificial stimuli. This is what we kind of expected to find more of based off of our expectation from the standard model, where, you know, regardless of whether it's the grading or the stem of the flower, we should be able to, you know, roughly be able to predict the response of that neuron. So these do exist, but it's only 10% of the neurons. And that, I think, was a little bit surprising to us, that it wasn't, you know, this is kind of what we really thought was going to be the majority of the neurons in the cortex. Finally, we can look at how these break down, comparing them across how these different classes are distributed across the visual areas and across our Cree lines. This is the plot I showed you a couple of slides ago, just how many neurons are in each class. I've just rotated it for space. And now we can look at these different classes across our visual areas. So that I think the kind of biggest thing that jumps out is that this none class, in black, goes from 20% of the neurons in V1 that don't respond reliably to any of these stimuli, is now 85% of the cells in RL. RL might be kind of special. There's some challenges with imaging in RL in terms of creating the retinotopic map. And so you might want to, you know, put a grain of salt, a little asterisk next to that, because there could be some experimental difficulties with that. But even in these other areas, AM and PM, it's almost 50% of cells that aren't responding to the stimuli that we're showing. The other thing that I find really interesting in this distribution, though, is that these blue bars here, these are the dark blue is the drifting gratings natural movies. This lighter blue is just natural movies. But these I think of as these motion selective cells. These bars are pretty consistent across these visual areas, excluding RL, right? It's about 25% of the neurons in V1, as well as 25% of the neurons in AM, right? And as the number of visually responsive cells is lower in these areas, AM and PM, that means that now these motion selective cells are like probably half of the visually responsive cells, maybe a little bit more. And so this suggests that there's kind of actually an increase in the motion information in these areas here, AM and PM. While there's a decrease in the spatially selective neurons that are well driven by the natural scenes and natural movies, where we see that this purple and pink group of classes gets smaller as we move away from V1. So we see kind of this enrichment of motion information. And it's interesting because you know, several slides ago when I was showing you that direction selective information actually gets weaker in these higher visual areas, but you know, there's a lot of motion information, but perhaps just less direction selectivity in that motion information. And then finally, we can look at this across the different Cree lines. This is just within V1, and look at, you know, how many cells fall into each of these classes within each Cree line. And again, you know, as we saw with some of the derived metrics, the biggest differences are what we see with the inhibitory populations. So for instance, our somatostatin cells here have almost no cells that don't respond to any of our stimuli. This black bar is very, very small. And in fact, almost half of the cells respond to all of our stimuli. Somatostatin cells have very robust, very reliable responses across all of our stimulus categories. We do get other cells in other classes, but by and large, these somatostatin cells really do respond really reliably and robustly. The VIP cells, I think, you know, from the literature, coming into this from the literature, I might have expected the VIP to have a lot more of these, of the none cells in the black bar. But really the VIP cells here in layer 2-3 and in layer 4 are dominated by this natural scene, natural movie, where the spatial statistics of natural stimuli are really effective at driving these VIP cells. And I think one of the things that we know about VIP cells is they have very strong surround suppression, which is why they don't respond to these large full field drifting gratings. But with the natural scenes where you don't have that type of full field consistent stimulus, we actually get really strong, robust responses. And so we see most of those neurons fall into this natural scene, natural movie category. There are some differences among the inhibitory cells. So there's, I can point out the FESF, this is one of our layer 5 populations, or the SCN1A, one of the layer 4, that do seem to kind of deviate from the other excitatory populations a little bit more than average. But by and large, our excitatory cells all kind of look the same as we saw earlier. So with that, I will conclude and wrap up. We see a lot of diverse and variable visual responses throughout the mouse cortex. 10% of our neurons look like what we expect from the textbook, look like that are well predicted by the standard model and respond reliably to all these stimuli, whereas 35% of cells don't respond reliably to any of these stimuli. And one of the really interesting questions that we are kind of grappling with now is what are these cells doing, these what we call the none cells. It's possible that it just has to do with the stimuli that are being shown. If we had included different spatial or temporal frequencies, or if we had a different set of natural images, that we would have evoked more responses from these neurons, and that's definitely a possibility. Another possibility is that these neurons, there's a lot of motion integration as well as multimodal sensory integration of different sensory modalities, and that these neurons kind of require other types of sensory or motor signals to really elicit activity from them. I mentioned, as I mentioned a couple times, the excitatory populations really show a lot of similarity in their functions, in their functional responses, while our inhibitory populations, these VIP and somatostatin cells, show very different responses, and that are indicative that they have very different roles in this cortical circuit. And so with that, I will just point you that there's a lot of information. All of the data is available through our website and through our software kit, the SDK. And so there's a lot of information about finding the data and using the data there. I'll point you, everything that I talked about today is from this paper in Nature Neuroscience. We also have a preprint that talks about the hardware for the pipeline. And then I'll just point you to another paper that I didn't have time to talk about that digs into these inhibitory circuits a little bit more and looking at those roles. So with that, I will thank the people that have worked on this. It's a very large team of people, and I'm just going to list a handful of people. Michael Bice and Jerome Lecoq are kind of the co-leads on this project with me. Jerome runs our optical imaging team that has collected all of this data. Gabe, Michael, Oliver, Dan, and Peter have been involved in analyzing these data. Pete Grabluski runs the neurosurgery and behavior team that does a lot of the early stages of the pipeline, but also has been very involved with developing a lot of the hardware for registering the pipeline. And then our informatics team, David, Feng, Lydia, and Wayne, have created our software kit and all of the tools for accessing the data and sharing the data. I also want to thank Paul Allen for his vision, encouragement, and support in creating our institute. And with that, I thank you for your attention and happy to answer questions.
Thank you, Saskia.
Yep.
Any questions can be typed into Q&A.
Let me see. I see this question here from Mario. Hi, Mario. In the model, did you try to let running speed modulating the responses with a multiplicative factor as opposed to being an additive variable? Okay, good question. And are preferred directions and orientations consistent between the drifting gratings and static grating cells? And the receptive fields from natural images consistent with the receptive fields from sparse noise. All right, for question number 1, so in the work that I just showed you today, we only had the running as an additive. We have other work. We had a poster up at SFN where we showed comparisons with multiplicative and additive, and we do see some differences. And I'm probably going to, I don't think I'm remembering this wrong, but we actually see that the model performance is better with it as an additive variable than a multiplicative variable. It's somewhat possible I just got that completely backwards, but I'm happy to send you some figures on that. If you want to shoot me an email, I can send you that. But we have done some of that comparison for sure. Are the preferred directions consistent? I haven't looked at this in great detail, so I can't say X% of neurons have the same. When I do look at it, they tend to be the same. Actually, no, I have looked at this in greater, again, I haven't quantified it, but they are highly correlated. It's not perfect, but they are pretty highly correlated. In terms of the receptive field center, we haven't done this with the receptive field with the natural images consistent with the sparse noise. The sparse noise, it varies a lot by which Cree line that we're looking at and which areas from. We don't get a lot of, we get fewer receptive fields with the sparse noise than we would have liked, or than what we'd like. And so it's, we could definitely do this analysis. I think it's a great question. But the number of neurons that we'd be using is a lot smaller than that then makes us happy. So we tend not to do those things, but I think it's a great question to think about. Thanks. Let me see. A question from you, how have you tried to measure the percentage of clusters with different reliabilities across all layers in V1? I'm not sure I understand this question. Yeah, I don't think I really understand this. So we've done this clustering analysis of, and we've looked at how the cells in V1 fall into these different classes of these different clusters that we find based off of their reliability. And so we've looked at this in that last slide I showed. We looked at this across all of the different Cree lines within V1. So we do, I guess I would say, yes, we have done this, unless you're asking something a little different, in which case, feel free to ask again. Question from Alexander. Do you have an estimate of what percentage of cells are not responsive to any stimulus in layer 2-3? Yeah, so we do have this, and so if in that last slide that I was showing, you could see that it was probably about 20% of the neurons in V1 layer 2-3 fall into that none category, where they don't have reliable responses to any of the stimuli. And then it gets larger as you move into other visual areas. But in V1, in the excitatory cells, kind of across all of the layers, it's about 20%, maybe a little bit less in layer 5. But yeah.
Any more questions? Okay, there's one more.
All right, Joram Kaiser, do you think it would be interesting to replace the use of a deep network to predict neural responses? Yeah, I do, and we've done some work with this, and actually, if there's a bioRxiv preprint version of our paper that actually had this, but we removed it from the final version of the paper, where we compared a couple of different CNNs to the activity of different CNNs at the different layers of the CNN to the responses that we have recorded at the different areas and layers of the visual cortex. And it's an interesting comparison to make. And so I do think that there's a lot of interesting questions to be addressed by this. And we've got kind of more work building more complex models. And I think this is in many ways kind of a lot of the direction that we want to go in, right? So I mentioned that this is a standard model that has existed in the field for many decades. And what we've done so far is basically say, this is how well the standard model does. It's okay. And for a particular group of neurons, it's pretty decent, but it doesn't really do a great job for these other neurons. And I think one of the challenges, both for us, but also for the larger community, because these data are available for everyone to use, is can we build better models? And one way of doing that might be using a deep network. And as the questioner points out, that they're harder to interpret, but I think there's ways to use neural networks to kind of think more deeply about kind of the types of transformations that might be happen. So yeah, I think that's definitely an interesting avenue of work. Erica asks, it feels like object recognition isn't the best task for the mouse visual system. Do you think there is a more appropriate task? I love this question. Yes, I do. So one thing to keep in mind when we think about the visual system in primates and cats, we talk about the ventral stream and the dorsal stream. And we believe that we have similar streams in the mouse. One thing to keep in mind is most of the visual areas that we reported from, the higher visual areas, would fall into the dorsal stream. And so, and that's because the areas that fall into the ventral stream are harder for us to access with the calcium imaging based off of our window configuration. And so it's possible that object recognition might be more interesting if we got more of those ventral areas. And when these experiments were designed, we were trying to get more of those ventral areas, but it didn't happen. So I think a more appropriate task would have a lot more to do with motion and navigation, especially in the dorsal areas that we're looking at. And so I do think there's a more appropriate task. I think it has to do with motion. And I think there's, we're only starting to think about really what a mouse is doing ethologically. I think we need to move away from thinking of a mouse as a little primate and need to start thinking about what its ecological niche is and designing our questions around that. So I think there's a lot that we can learn in taking that approach. Yeah, thanks for that question.